LPA principle and technique. LPA principle. Line probe assays are a family of DNA strip-based tests determining the drug-resistant profile of a MTBC or Mycobacterium tuberculosis complex through the pattern of binding of amplicons or DNA amplification products to probes, and targeting the most common resistance associated mutations to first and second line agents and to probes targeting the corresponding wild type DNA sequence. Mutations are detected by the binding of amplicons to probes targeting the most commonly occurring mutations probe, or inferred by the lack of hybridization, that is, lack of binding of the amplicons to the corresponding wild type probes. The post hybridization reaction leads to the development of colored bands on the test strip detecting probe binding. LPA technique. DNA is extracted using Genolyze version 1 kit and amplified using a PCR machine. After chemical denaturation, single stranded amplicons bind to the membrane strip. Membrane strips are coated with specific probes complementary to the amplified nucleic acid. Highly specific binding of complementary DNA strands is ensured by stringent conditions which result from the combination of buffer composition and a certain temperature. Thus, the probes reliably discriminate several sequence variations in the gene regions examined. The streptavidin conjugated alkaline phosphatase binds to the amplicons biotin via the streptavidin moiety. Finally, the alkaline phosphatase transforms an added substrate into a dye which becomes visible on the membrane strips as a colored precipitate. A template ensures the easy and fast interpretation of the binding pattern obtained. Pros of LPA include less turnaround time or rapid results, specific information, less biohazard risk involved in comparison to conventional methods, may provide information on virulence, may provide information on the level of resistance, and may help to identify molecular pathways to develop new drugs. Cons. Some mutations that confer resistance are outside the regions covered by the test, and therefore resistance cannot be completely excluded even in the presence of all wild-type probes. Thus, in some cases, additional phenotypic DST may be necessary to provide full assessment. Some mutations are identified specifically by mutation probes, whereas others are only inferred by the absence of binding of the amplicons to wild-type probes. The lack of binding of a wild-type probe without simultaneous binding of a mutant probe is likely caused by the presence of a resistance mutation. However, systematic errors are possible due to synonymous and non-synonymous mutations, which is globally rare, but these isolates can be frequent locally. LPA is less efficient than conventional culture-based DST in finding resistance in samples harboring both drug-susceptible and resistant bacteria, that is, heteroresistance. More specifically, with LPA, it is possible to detect resistant bacteria with mutations detected by the mutation probes if resistant bacteria represent at least 5% of the total population. However, resistant bacteria with mutations inferred by the absence of wild-type probes would probably be missed if the resistant population is less than 95% of the total bacterial population.